The cell cycle is a series of events that takes place in a cell leading to its division and duplication. Interphase is the phase of the cell cycle in which the cell spends the majority of its time. It is divided into three stages, G1, S phase, and G2. In G1, protein synthesis occurs and the cell doubles in size. In S phase, chromosomes located in the nucleus are duplicated. In G2, the cell resumes its growth in preparation for mitosis. After interphase, the cell begins the meiotic phase. The first stage of mitosis is prophase. In prophase, chromosomes become more coiled and can be viewed under a light microscope. Each duplicated chromosome appears as a pair of sister chromatids, joined together by their centromeres. In the cytoplasm, the meiotic spindle forms between the two pairs of centrioles as they migrate to opposite ends of the cell. The nuclear envelope disappears at the end of prophase. This signals the beginning of prometaphase. In prometaphase, the chromosomes are now highly condensed and each sister chromatid has a kinetochore, a specialized protein structure located at the centromere. Some of the spindle microtubules attach to kinetochores, moving the chromosomes into an agitated motion. Other spindle microtubules attach to opposite poles, moving the chromosomes towards the center of the cell. Metaphase is the longest stage of mitosis, lasting about 20 minutes. In metaphase, the centromeres are at opposite poles of the cell and the chromosomes are now lined up on the metaphase plate. Anaphase is the shortest stage of mitosis, lasting only a few minutes. In anaphase, the cohesion proteins or centromeres are cleaved and the two sister chromatids suddenly move apart. At this point, each chromatid becomes its own chromosome. These two free chromosomes now move towards opposite ends attached to the microtubule, leading to the centrosome. By the end of anaphase, each side of the cell has completed chromosomes, meaning full parental DNA and the same number of chromosomes. Telophase and cytokinesis are the last stages of mitosis. Two daughter nuclei form in the cell. The nuclear envelope arises from the fragments of the parent's nuclear envelope and other portions of the endomembrane system. The nuclei also reappear. Cytokinesis takes place late into telophase. A cleavage furrow develops and pinches off the two cells.
Meiosis is cell division that produces reproductive cells in sexually reproducing organisms. The nucleus divides into 4 nuclei, each containing half the chromosome number. Meiosis 1 begins with prophase 1. During prophase 1, the chromosomes begin to condense and the homologous chromosomes loosely pair along their lengths, aligned gene by gene. Crossing over, or the exchange of corresponding segments of DNA molecules by non-sister chromatids, occurs and is completed during synapsis. Synapsis ends mid-prophase, and the chromosomes in each pair move slightly apart. Each homologous pair has one or more chiasmata, points where crossing over has occurred. Centrosome movement also occurs during prophase 1, and the spindle begins to form. Metaphase 1 follows prophase 1. In metaphase 1, the pairs of homologous chromosomes are now arranged on the metaphase plate, with one chromosome in each pair facing a pole. Both chromatids of one homolog are attached to the kinetochore microtubules from one pole. Those of the other are attached to microtubules from the opposite pole. In anaphase 1, the breakdown of proteins responsible for keeping the chromatids together are separated, allowing the homologs to separate. The homologs move toward opposite poles, guided by the spindle apparatus. Still, though, sister chromatid cohesion persists at the centromere, causing chromatids to move as a unit toward the same pole. At the beginning of telophase 1, each side of the cell has a completed haploid set of replicated chromosomes. Each chromosome consists of 2 sister chromatids. One or both sister chromatids contain segments of non-sister chromatids, causing variation. Cytokinesis occurs and a cleavage furrow develops separating the 2 haploid cells. In some species, the chromosomes condense and the nuclear envelope reforms. Meiosis 2 begins with prophase 2, where the centromere replicates into 2. The nuclear envelope, if present, begins to fragment. The 2 sister chromatids start moving towards the metaphase plate. In metaphase 2, the chromosomes are positioned on the metaphase plate as in mitosis. Due to the crossing over in meiosis 1, the 2 sister chromatids of chromosomes are not genetically identical. The kinetochores of the sister chromatids are attached to microtubules extending from opposite poles. In anaphase 2, the breakdown of proteins holding the sister chromatids together allow the chromatids to separate now, becoming individual chromosomes. These chromosomes move toward opposite poles in the cell. In the last stage of meiosis 2, the nuclei form and the chromosomes begin to condense. Cytokinesis occurs late in telophase 2, and cleavage furrow separates the 2 cells. As a result of meiosis, 4 daughter cells are produced, all genetically different from one another.